Okay, welcome. Now this lab is going to cover the brain and the cranial nerves. We've already did the spinal cord, and we've already completed certain other neural topics. I want you to also be aware that there's a few of these sections that we've just decided for one reason or another that we can't use. This will include exercises 14.1 to 14.3. As well as 15, but there are some sections that are omitted. So I will specify those. There will be a lab worksheet hand-in. And as always, these hand-ins are due by 5 p.m. on Friday. So let's take a look at this and we're able to explore further the brain and the cranial nerves. Now our objectives are to review the exercises 14.1 to 14.3, 15, and to use the nervous system where we can see some of the slides and the brains and the diagrams. Now, some of this is going to be, if we are not in class, supplemented by videos. So I encourage you strongly to take time to review what's in your book, as well as microscopic images from the PowerPoint and from the lab videos. Now, we're not going to do the sense organs this time. We're going to actually go into a further lab on special senses. But you will have a brain review model that probably will be demonstrated in a video. And you need to also record any information in the lab exercise sheets. Now, you will complete the review sheet at the end of exercise 15. That should be pages 275 to 278. Take time to review the videos of the dissection of the sheep brain. This will help you to comprehend the neuroanatomy. Now, again, if you're not doing this in lab, we got to show you what's really going on. And that shouldn't be it shouldn't be difficult. Okay, let's get moving. Now, if we start out on 14.1 to 14.3, what are we looking at? In the brain, we have a variety of cell types that make up nervous tissue. The two cell types best known, neurons, and they're classified by structure and function, but these are the ones that basically communicate using action potentials. Glial cells, these provide for a variety of functions that protect and support neurons. They do not provide action potentials, but they keep the conditions proper so that the neurons can function. Just one pointer. Astrocytes, for example, which is a type of glial cell, these control the amount of potassium that is in the interstitial fluid, etc. I know that we've been teaching that potassium should be in greater concentration inside the cell than out. That's true. But because you're going to have a certain amount of fluid and a lot of neural activity, glial cells, there are astrocytes, tend to add an extra buffer of control to modulate the potassium ion concentration so it doesn't build up in any area to a significant degree. As we said, neurons are classified on the basis of structure and function. You're going to note here that the classification is going to be based on whether they have an axon or not. This is for neurons. Anaxonic means that there's no axon. Lots of dendrites found in brain and special sense organs, but no real axon to speak of. Unipolar, these have dendrites and the axon, and they're kind of like fused together. The actual soma, the cell body, is kind of put on the side. You'll see this in a minute. This is where you have most sensory neurons are unipolar. Bipolar, the dendritic processes branch at the distal tip. The cell body is between the axon and the dendrite. This is kind of a rare situation. Now, by the way, hint, I want to give you just one point here. Never get bipolar neurons confused with bipolar, the mental disorder. Okay, no pun is intended. It's just sometimes students can do that, and I want to avoid that. You will see these special bipolar neurons in the organs that deal with sight, and it's supposed to be smell, not small, smell and vision. Finally, multipolar. These are where you have two or more dendrites and a single axon, and they are most common in the CNS and found in all motor neurons. So they have some role in the initiation of contractions in skeletal muscles. Here is anaxonic. As you can see, lots of dendrites, but no clear start point for an axon. Here is the unipolar. As you can see, really what's going on is you've got this extension on this side and extension on this side. The bipolar is interesting also because it's similar. Unipolar, the cell body still has to go through this, and then the axon comes down, then you have the axon terminals. But notice where the dendrites are. They have sort of a lengthened process, and they are arrayed at the top here. This is where you're really going to have communication from other cells. They are going to have synapses here. Now, with bipolar, notice that you have two axons. And maybe that's one of the distinguishing factors. Also notice that the cell body is kind of pushed to the side. There's sort of an extension here. So you have axon from here, from the dendritic initial segment is right here. So that means that action potentials start here and go all the way down. And then you have axonal terminals here. Okay? If you have problem getting these two confused, think of it this way. Unipolar has one pole. If you want to say the axon would be the pole. This is not an axon. When you have bipolar, you have an axon up here and an axon down here. This is still the receptive area. This is still the communicating area, meaning that they would have synapses with other cells. And then we have the multipolar one, where you have dendrites extend out around the soma, and then a very long axon, and then the axon terminals here. Now this brings us back to the graphic organizer that I provided for you last time. Keep in mind that it's a really simple deal when you're dealing with nervous tissue. Question, does the cell produce action potentials? Yes, then it's a neuron. If it's no, then you start going through this yes-no process, this almost dichotomous key approach. Does the cell form myelin coverings around the neurons? If it's no, then you start getting into these other unique cell types. Does the cell wrap around brain capillaries? Yes, astrocytes. If it's no, does it act as an immune cell? Yes, it's microglial. If it's no, it is ependymal cells, and you'd only find those really in the ventricles. If you go back to, does it form a myelin covering around the neurons? If it's yes, is the cell present in the central nervous system? If it's yes, it's oligodendrocytes. If it's no, it's Schwann cells and satellite cells. When we talk about the neuroglia, though, of the CNS, you have to have a greater appreciation for what all these structures are doing. Because when you think about it, you have to have in a brain the capability to process sensory information, to process all sorts of other components and factors, and then elicit a response. Now, processing in the beginning is your sensory sites in the brain. Making some intuitive decision, that may be what you might consider the frontal lobes, etc., perhaps connected with some of the emotions. But then you have the motor areas. Okay, so that's where you're going to get into your motor cortex. And they will control the responses, okay, whether it's glandular or muscular, etc. But let's take a look at these cells. You have the ependymal cells. They form a simple cuboidal or columnar epithelium that lines the cerebral spinal fluid, CSF, filled areas of the brain. That's basically the ventricles. You will also see, to a degree, fluid-filled areas in the spinal cord. Now, what ependymal cells do is they assist in producing, monitoring, and circulating cerebral spinal fluid. Microglia, these are developmentally related to the monocytes and the macrophages. Now, monocytes and macrophages, if it sounds familiar, these are cells that go out there and attempt to clean up after dead cells, such as an infection, or actually kill infective cells or infective pathogens that are present. So what microglia do is they will migrate into a CNS area during formation and persist as guard cells to remove waste, cellular debris, and pathogens by phagocytosis. All right. Astrocytes, these are the very interesting and unique cells because they form what is called the blood-brain barrier. They have these extensions, which they call pseudopodia, which kind of wrap around the capillaries. Now, what you're doing is you're creating several layers here to control what goes from the blood into the brain tissue and from the brain tissue into the blood. So in essence, they isolate the CNS. And it's not just isolating it from, let's say, bad things. Chemicals, hormones. They will regulate gas, nutrient, and other substances in the interstitial fluid surrounding the neurons. They will also form scar tissue after CNS injury. This is where you get the formation of things like lesions or plaques, where the cells have been damaged and where they don't get replaced. One of the interesting things to keep in mind with all neural cells, when neurons die, they're normally supposed to be living forever, and they don't necessarily undergo mitosis a lot after, let's say, birth, and even before that in some cases. So you can't grow new brain cells, okay? And so then everybody chuckles and says, well, what about when somebody studies and learns stuff? Well, that's forming new connections, new synaptic connections between cells. Now, we have oligodendrocytes. Now, these are the guys that help provide myelin sheath. They provide structural framework within the CNS by stabilizing the positions of the axons. Oligodendrocytes have extensions of their cell membrane, and they wrap around the axons of the neurons. The importance is that they are providing myelination. Myelin enhances the speed of action potentials, APs, in which the AP will jump from node to node. Each of these nodes are gaps where myelin is absent. It's also referred to as the nodes of Ranvier. Now, the regions dominated by the myelin axons are referred to as white matter of the CNS. And if you look at the brain, you will see that there are areas of distinctive, almost like color, but it's basically either kind of a grayish matter or a white matter. White matter is heavily laden with myelinated axons. The unmyelinated neurons, they're not covered by myelin or neuroglial processes, and they make up what we call the gray matter of the central nervous system. Here's an example. Now, they took this out of the spinal cord, but part of this is still applicable. If you notice, here we have astrocytes, here and here. And you notice they have these extensions that kind of wrap around capillaries, and that's your blood-brain barrier, okay? And here you have them, and some of them are also extended into other parts. You have this microglia cell right here. Notice it has sort of spiny extensions here. So they're also referred to as the spiny cells. These cells, as well as the astrocytes, will multiply after, let's say, some of the brain tissue has died, okay? Usually it's the microglia that will phagocytize any dead matter and then remove it. So We've got oligodendrocytes. If you notice here, this is how you get a lot of myelination. The cells will extend a tremendous amount of their cell membrane and then wrap it around several times, as you can see here. And so this myelination of the axons enhances the speed of signals processing. But instead of the AP, the action potential traveling down the length of this, it's going to literally jump from one site to the other, these nodes that exist. Now, neuroglia of the peripheral nervous system, you need to be mindful of this. The Schwann cells, otherwise called the neurolemocytes, cover peripheral axons in two different ways, as well as participate in axon repair after injury. Satellite cells, they kind of wrap around the peripheral cell bodies. Their function, not everybody is 100% sure of them. What they do know is that Schwann cells are important. When you have myelinated cells, the outer surface of the Schwann cell is called the neural lemma. Schwann cells protect the myelinated and unmyelinated axons from contact with interstitial fluid. Unmyelinated cells have Schwann cells that stabilize the position of the axons in clusters. This sounds similar to what you see with oligodendrocytes. Oligodendrocytes, though, only work in the CNS, not the PNS. Schwann cell plasma membrane wraps around an axon like a jelly roll, just wraps layer upon layer upon layer. But as it's wrapping, the cytoplasm that is inside of that membrane gets kind of squeezed out. So all you have is really just cell membrane wrapping around and wrapping around. And this is referred to as an internode. So here is my internode here. This is the neural lemma here. This is, of course, these are all Schwann cells, by the way. Notice that the nucleus is kind of stuck hanging out here, okay? And this is the axolemma. This is the axon, which is the cell membrane of the axon. And look at that, layer upon layer upon layer of this myelin sheathing. Here, as I said, the internode, and the area basically with no myelination, that's the node. So if you start the initial segment and you just had an unmyelinated axon, it would go, traveling down this route, going all the way through this length of axon. But if you have it myelinated, it would jump from here to here to here to here to here, jump over to here, jump over to here, and it would keep going. And in this way, the speed of action potentials is greatly enhanced. Now, if we have an unmyelinated axon, here's another example. If you have the Schwann cell, you have all these axons. They're not fully wrapped and myelinated, but they're kind of held together here, or sort of trapped. And this gives sort of an integrity and prevents from shifting within the tissues of the body. Now, when you take the time to do the central nervous system, you're going to see the microscopic examination of the cerebrum. It has gray matter. That's unmyelinated neurons. It has white matter, myelinated axon. The cortex of the cerebrum is basically gray matter. To help you to understand this, when I'm talking about the cerebrum, if you compare cerebrum to, let's say, the spinal cord, okay? Cerebrum, the outer edges are gray matter. The inside is all white matter, which only makes sense because, hey, if you're processing signals and you're doing various functions, you're going to have your wiring on the inside communicating with one section and other sections of the brain, etc. And we'll get more into this when we get into the lecture. Now, with the spinal cord, you have all of this heavy insulated fibers that travel up to the brain, down through the body, etc. And you have some of the unmyelinated cells in sort of the core or the center, okay? If you take a magnification here, you're going to see what they call a pyramidal cell. Now, here's the point of a pyramidal cell. You will see, and this is through special staining, these extensions here, which are sort of the, what you might say is dendritic approach, which is connecting now to various other neurons in the area, a relatively mild or short distance axon, which then has basically its terminal axon terminals down throughout here. Okay, to help you understand how important this was, it took a lot. Emilio Golgi was the one that came up with, yes, he also came up with Golgi apparatus, but he developed a special silver stain that only partially stained some of the cells, and so it allowed you finally to see brain tissue and the individual cells, such as you see here. Now, when we get to the central nervous system, the microscopic examination of the cerebellum, and it's easy to get confused. Think of cerebellum as the small brain, cerebrum, big brain, okay? The layers. Now, with this cerebellum, this is extremely important structure. This is how you're able to walk a tightrope. This is how you're able to juggle. This is how you're able to balance and skate or ski or do anything that needs coordination, balance, etc. The white matter that you're going to see, there's a major vein of it. Well, I shouldn't say vein, but massive amount of it. And it's called the arbor vitae, which means tree of life. And this is all myelinated axons, carrying various bits of information back and forth from the rest of the body to the cerebellum and back again. There is a structure called the molecular layer. These are interneurons that interconnect with other neurons in the cerebellar cortex. Then you have a Purkinje layer. This is a thin layer that contains multipolar neurons whose axons serve as a major output channel to transmit information from the cortex to the deeper cerebellar nuclei. Sounds like a mouthful. Here's the point. How are you going to communicate with the cerebellum with the rest of the brain to coordinate? So instead of just tossing a ball flippantly, you're able to smoothly throw that ball like a pitcher on a baseball team. It takes a lot of muscular coordination. And they're constantly getting feedback going, okay, too much, not enough, too much, not enough. Okay, it's right. Okay, got to move this muscle this way. And we have also a section of this tissue called the granular layer, which includes neurons that receive most of the information coming to the cerebellum from other brain areas. So here you have the molecular layer here, the granular cell layer, the Purkinje cell layer, which is kind of this thin layer here, and then you have white matter. So you think about it this way, these are the cell layers doing a lot of complex processing. And from that, you have the information coming up through here by the white matter and then leaving by the white matter. Now, there is a couple things I want you to be aware of. In this case, we would have normally dealt with dissection of sheep brain. The meninges, depending on where you get the sheep brains from, are either easy to see or they're somewhat tough to see. I'm going to try to include a nice little video in the lab videos for you to view this. If you don't have the opportunity, such as with an online presentation, you'll be able to have some appreciation of this now and in some videos. The thing that you want to keep in mind is this: how do we protect the brain? Well, there are three connective tissue layers that protect the brain. They are the Dura Mater, the arachnoid matter, and the pia mater. Now, Dura Mater has two layers: an outer endosteal layer, which is fused to the periosteum of the cranial bones, and a meningeal layer, which forms the brain covering. There is an arachnoid matter. Now, just below the arachnoid matter, you're going to find all these little tissue folds and little fragments. And what it's interesting is they kind of form this almost like web-like appearance. That's why they call it the arachnoid. And in that spacing also is CSF, cerebral spinal fluid. With pia mater, though, it's thin, but it's very tightly attached to the brain tissue itself. Now, there are also several dural sinuses. These are separations of the Dura Mater, where drainage of venous blood from the brain to the internal jugular vein occurs. This is kind of important because, hey, if you make too much cerebral spinal fluid, you've got to drain it somewhere. And we're going to talk to you about that drainage. Now, the folds of the Dura Mater form partitions between major brain structures as well. This allows you to have anchoring of the brain to the rest of the skull. The dural folds include what's called the falx cerebri, which are folds into the deep groove, that is the longitudinal fissure, and they separate the cerebral hemispheres. The tentorum cerebelli separates the cerebrum and the cerebellum. So there's a little pocket in there, and so this tissue gets in there. Think about this. The brain, not the preserved brain, but the brain itself is very, very fragile. Okay? It doesn't have bones. It doesn't have lots and lots of cartilage. And the worst thing you could ever do is have the brain bouncing inside the cranium. That's the worst thing. That's where you end up with concussions and all the other things. So you have some anchoring, and you have some means using the buoyancy of the cerebral spinal fluid, which would act as cushioning. And then you have these other layers that help to protect it. Let me finish my dural folds. There's also falx cerebelli, which is separated cerebellar hemispheres. Now, we're going to move through some of this a little bit quicker, but I'm going to try to give you some images to support what you're going to be seeing. If you looked at the sheep brain, and the sheep brain is used, by the way, just because it is very easy to get sheep at the slaughterhouse, the abattoir, and they put these sheep brains aside for scientific review. Arachnoid matter has thin filaments that I was talking about earlier that cross the subarachnoid space, the space between the arachnoid matter, and then it comes down and attaches to the pia. The subarachnoid space has cerebral spinal fluid circulating it. I mentioned that. Sheep brain sections that you want to be able to observe, and when you see the videos of the dissection, review it. A lot of times the dura mater is there, sometimes it's not. But no matter what, if you were to have one in hand, you have to eventually move this kind of thickened layer off so you can basically view the brain. You will also see on the underside, the inferior side of the brain, the ends of the optic nerves, which cross over at the optic chiasm. We have the cerebral hemispheres, which are separated by the longitudinal fissure. The lobes of the brain include the frontal, parietal, temporal, and occipital. You have also the longitudinal fissure, as I mentioned to you before. And then when you start looking at some of the finer details of any section of the brain, you're going to see gyri, sulci. Gyri are the ridges of tissue. Sulci are the shallow grooves. To help you to think about that, because everybody sits here and goes, so what? Well, think about it this way. If I take a straight piece of paper, I only have so much surface area. But if I start folding it, creating these ridges and then these grooves, I can put together a lot more surface area where you just form these, if I want to say it this way, these mountains and these valleys, these mountains and these valleys, these mountains and these valleys. And all the mountains are brain tissue, okay? If you need to seriously separate one main structure of the brain from the other, that's where you start having fissures. These are deeper grooves, okay? Also, that when we look in a brain, and we look at the sheep brain, for example, you're going to see, of course, the cerebellum. You also have the transverse fissure. Remember, this is the fissure that kind of separates cerebellum from the cerebrum. The corpora quadragena. Now, interestingly enough, if you were to pull back on the cerebellum, you will see these structures on the midbrain. Corpora, bodies, quadragema, basically 4 units. So these 4 units consist of 2 that are larger, like bumps. superior colliculi, and those are involved with controlling the visual reflexes. The inferior colliculi are right next to them, but below them, and they're sort of smaller little bumps. These are the provide for the auditory reflexes, this inferior colliculi. If you look on the underside again of the sheep brain, you would see the olfactory bulbs and the olfactory tract. The tract is easier to see. The bulbs aren't always guaranteed because, in part, in the slaughterhouse, they're not gentle with removing these brains. They're in production, so they've got to pull them out, and they don't always come out with everything necessary. So you get them, sometimes the bulbs I've seen torn away, they're torn in part, etc. When you go into the intracranial part, you will also find the pituitary gland, but that's a possibility. Or you will just find the stem, which is infundibulum. What happens if you remember your neural cranium of the skull? We have this saddle-like structure, the sella turrica, and that's where the pituitary gland is settled in there, nested in there. When they pull the brain out, a lot of times what is left behind is the pituitary gland. Finally, we have the brain stem, which is divided into the midbrain, the mesencephalon, the pons, and the medulla oblongata. Here is the gyri, the rising areas, and here are, of course, the sulci. Here's the parietal lobe. Here's the frontal lobe. Here's a transverse fissure, and then, of course, here's the cerebellum. Okay. If you look at another image, and you can see this, you can see clearly, and by the way, don't panic. I won't necessarily sit there and say, tell me where the abducens nerve is. What happened was, when these brains were taken out, a lot of the cranial nerves get frayed out. The only ones that are basically reliable at all for identification are the optic nerve, which would be for the eyes, and the olfactory bulb or the olfactory tract, which would be the sense of smell. Okay? Right around here is where you would have normally seen, the infundibulum is right here, and right on top of that would have been this P, kind of rounded little shape, and that would have been the pituitary. Okay? Here is the optic tracts. Okay? And you have this bump here called the mammalian body. Trigeminal nerve here, the spinal cord is here. And you can see some fraying here of some smaller nerves. Right here, this kind of rise here is called the medulla oblongata, and then the next one over here is the pons. You have the cerebral peduncles. These are structures that eventually attach to the cerebellum. Okay, and what they've done is they've removed some of the cerebellum here from what you can tell, or it's actually partly it's going to be over on the other side here. Here is the Oculomotor Nerve. Like I said, I'm not going to hold people on some of that. Here is the cerebellum. This is the transverse fissure. Now, if you were to put pressure and almost like beginning to bend, putting one hand right here and one hand right here, in here you would see the corpora quadrigemma. This is the brain stem. This is the spinal cord. And what they did was they peeled away some of the meninges so you can see how they are. They're kind of like somewhat translucent, but they're very tough. You'll see the sulcus and the gyrus and the cerebrum. Now, if you were to do this as a dissection, sheep brain mid-sagittal sections provide internal and anterior to posterior view of most of the major sections of the sheep brain. You'll see an image of that in a second, but you would be trying to locate and understand the function of the following structures. The cerebrum, which interprets sensory input, controls voluntary motor activity. The corpus callosum contains myelinated nerve fiber tracts that connects the two hemispheres together. The fornix is a fiber tract that connects the mammillary body to the hippocampus. The cingulate gyrus, part of the limbic system that regulates emotions and other related behaviors. Then we have at the far end, as you look inside, the pineal body. Now, the pineal gland, or the pineal body, secretes melatonin, and this is involved with the sleep-wake cycle. You would also be able to locate the thalamus, which acts as a relay station for sensory impulses. Think of it this way: if I have all this sensory information coming up from my body, whether it's heat, pain, pressure, whether it's at the same time also things like sound and smell and taste and everything, it all comes up to this area of the thalamus and then gets reprocessed to specific lobes of the brain. Visual goes back to the occipital cortex, the hearing goes to the temporal, and you can go on and on and on with that. The somatosensory part is in the parietal. Now, as I said to you before, the cerebellum, this regulates motor activity to control timed and smooth muscle motions. What I mean by timed? Well, think about it. You have the professional skaters that suddenly start spinning around and they know how they've been able to control all their muscular moves to accelerate their speed as they spin, and at the same time controlling their body and flexing it so that they can achieve the maximum speed as they're spinning. And then they also know when to stop, which is pretty cool when you think about all the motions, and they're keeping balance at the same time. Now, the hypothalamus, this is extremely important, and they consider this now more or less the master gland. It regulates autonomic nervous system, and it regulates pituitary gland secretion, controls body temperature, food and water intake. There's a structure called the mammalian body, which is part of the hypothalamus, and it's part of the limbic system and controls motor reflexes associated with eating. The midbrain contains the superior and inferior colliculi. The pons contains fiber tracts that connect the cerebrum to the cerebellum. Okay, this is the peduncles that we were talking about. It also has fiber tracts that connect to the medulla oblongata, the spinal cord, and centers for regulating the breathing rate. Okay, now what I missed here, and the water intake in mammalian body. Okay, and we move forward, and then we have the medulla oblongata. Sorry about that, a little extra slide there. This contains control centers for regulation of heart rate, breathing rate, and blood vessel diameter. Okay, think about it this way just briefly. Blood vessel diameter. If the diameter increases, the flow of blood will increase, yes, but the pressure will go down. If you reduce the diameter, the flow may increase, but it will be under a greater pressure. Just as if you take a garden hose and you squeeze it, it comes out faster. Okay? That's what happens sometimes when the body needs this pressure elevation to get the blood moving to a certain area. It may vasoconstrict as opposed to vasodilate. More later on that. Okay, let's take a look at some of these things. Here's our medulla. Here's our spinal cord. Here is this cerebellum. And you see all this white area here. This is the arbor vitae. So there's a lot of gray area here. And that's necessary for all of this information processing of coordination and balance and control of movements, etc. But this information gets fed back, comes out, etc. And therefore, you're going to have heavily myelination here to speed up the AP, which is going to carry the information of position, etc. Now, we talked about the superior and inferior colliculi. Here's the inferior colliculi. Here's the superior one. They're big. We have the pineal gland here. The hypothalamus is down here. Now, hypo meaning also below the thalamus, and the thalamus is up here. The hypothalamus is down here. Okay, you have the corpus callosum. You notice how it's white? Heavily myelinated. It's got to pass information from left hemisphere to right hemisphere and back and forth. We have the gyri up here. We have the sulci. Okay, so the sulci are these shallow indentations. The gyri are these raised structures of uh, nervous tissue. Okay, here's another dissected brain where they went mid-sagittal in their cut. You can see the corpus callosum here. You can see also the lateral ventricle here, this space. This would normally be filled up with CSF. And you have here also the third ventricle, because the lateral ventricle empties into the third ventricle. You have this tissue here called the fornix. The pineal body is going to be right back here. The arbor vitae of the cerebellum is here, the medulla oblongata, and then of course, we have the superior and the inferior colliculi. Now, a coronal section is used to observe the organization of gray and white matter in the cerebrum. Usually this includes a deep gray matter nuclei. So what you're going to do is when you take and do a slice through this coronal section, you should be able to notice the following. Again, I want you to understand a lot more of the function than in some cases, I'm going to give you a picture, a blurry picture, I'm not going to do that, of the brain and say, okay, where's what? The corpus callosum, as I mentioned, is fiber tracks between the two hemispheres. So that's how the left brain communicates with the right brain and the right with the left. The cerebral cortex is the thin layer of gray matter on the surface of the cerebrum. The white matter is deep to the cerebral cortex. This is all that heavily myelinated structure, all the fine wiring connecting different parts of the brain. The basal nuclei, these are interesting because when you have nuclei, you have clusters of cell bodies. So they're going to appear like gray matter surrounded by all sorts of white matter. And so this is going to be deep to the cerebrum. They regulate the intensity and the degree of voluntary movements by adjusting muscle tone. Lateral ventricles, these are deep within the cerebrum and they contain CSF. The septum pellucidum is a partition that separates two lateral ventricles. The internal capsule is a band of nerve fiber tracts of white matter that travel between the thalamus and the basal nuclei. So there is means to pass this back and forth. So what do? Here is our coronal section. Here is our internal capsules here. Here is the fornix. This is the lateral ventricle here, okay? And what you have here is the corpus callosum right here, kind of attaching and connecting to this side of the brain and that side of the brain. Cerebral cortex, here, 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 here. All of that, that's much more darker, okay? But you notice that there's a lot of myelination here, which means there's a lot of pathways connecting to different parts of the brain. There are a couple of other very special parts of these nuclei. The caudate nucleus is here, the putamen here. They both make up what's called the basal nuclei. Here you have what is referred to as the diencephalon. The diencephalon is really consisting of the thalamus, the hypothalamus, the epithalamus, which is a little bit above, and part of it at one end has the pituitary gland. Here is this separation here called the septum pellucidum, all right? So these are the lateral ventricles, and they eventually come down to eventually fuse and empty into the third ventricle. Now, as I mentioned before, I've talked about ventricles. Let's go a little bit more into those. These are a series of interconnected deep cavities derived from the internal space of the embryonic neural tube. Ependymal cells, these basically line the ventricles, and the space is filled with cerebral spinal fluid. You want to note the diagrams that you would see in the Siracus book, pages 260 and 261. When you're doing that, locate the lateral ventricles. These are deep structures within the cerebral hemispheres. The interventricle foramen connects the lateral ventricles to the third ventricle. The third ventricle is positioned between the two thalami in the diencephalon. Now, there is a drainage port called the aqueduct of the midbrain. It connects the third to the fourth ventricle. Now, the fourth ventricle is anterior to the cerebellum, okay, and extends into the medulla oblongata, and the flow then enters the central canal of the spinal cord. What am I saying here? Here is the cerebellum. Here, over on this side and on this side would be the lateral ventricles. They empty into this third ventricle. There's a tissue that connects both sides of these, that this sort of runs in between it. Then it empties into this very interesting little cerebral aqueduct, and then comes and forms the fourth aqueduct. Now, the fourth has several apertures or outlets. The lateral one's on the side, the medial one in the middle, and basically you're going to have cerebral spinal fluid circulate around and then up. This is the superior sagittal sinus. This is this massive section where a lot of venous blood flow goes. But here's the point. CSF was partly created by the choroid plexuses here, and there's another set of choroid plexus here. Sort of like a nice little tuft of capillaries, but wrapped with the ependymal cells. So we can't have the fluid just being made forever. It has to be reabsorbed into the blood. That's where we have these little arachnoid granulations. They're usually up in here, and basically it's like a one-way valve. CSF goes into the blood, nothing comes out, backflow. Now, one of the other things is that you will have the central canal of the spinal cord having this run all the way down, and of course you have also fluid following around the spinal cord. Okay? The spinal cord has a continuation of its Dura Mater, then the arachnoid matter, subarachnoid space, and then of course you've got the pia mater tightly up against the spinal cord. Everybody with me? When the spinal cord ends, it comes down to this section we call the cauda equina, and then of course this fibrous connective tissue here, the filium terminae. You notice that the fluid goes down, up, around, all over the place, but then eventually it circulates up to be reabsorbed up through here. Okay? Now, the choroid plexus are a capillary network in close connection with the modified ependymal cells. Cerebral spinal fluid forms from blood plasma, which is filtered through the ependymal cells. Here's the point. You mean it's just blood plasma? No. It has an altered ion concentration from blood plasma. Keep in mind that blood plasma, and this is when we've spun off all the red blood cells, white blood cells, platelets. This plasma is sort of straw-colored. It has a lot of water, ions, sugars, various hormones, some antibodies. You will have a few other proteins of this, that, and the other thing. It will have lots of different ions, electrolytes. The concentrations are different as this plasma gets processed and filtered through the ependymal cells. And that's what truly makes CSF. It's different from plasma. The CSF drainage occurs at the arachnoid granulations. These are really extensions of the arachnoid matter. They project into the dural sinuses, and the CSF filters through these granulations, is drained into the venous blood flow, as I've mentioned to you before. Here's where we start, blood coming through. Here's the interstitial fluid in the thalamus. And we've got this nice choroid plexus sitting around here. The fluid will attempt to pass through here, but here's the problem. The ependymal cells will be picky. They will control what is allowed in and out. So now we have something with a different chemistry from blood plasma circulating around here. and that's the CSF fluid. Now, we eventually have to deposit this back into the blood, and this is where, here's the arachnoid matter, the subarachnoid space, and you'll notice that these granulations are here, and this is sort of the structure. Even though you've got venous flow coming through here, this flow is going to be greater and it will seep out back into the venous blood, okay? Pia mater's here, cerebral cortex. You have this subdural space here, and all this fluid is going to just leak out. Nothing comes back in. That's what I want you to be aware of. Now, I dropped exercise 15.6 because we don't have a biopack set. No problem. Here's 15.7 that I do strongly encourage you to spend time on. Now, it's one thing to kind of just say, okay, olfactory, optic nerve, oculomotor nerve, trochlear nerve, trigeminal nerve, memorize it, set it to a mnemonic, etc. Those are helpful, but you got to know what they do. Key points. The cranial nerves are 12 pairs of nerves. They are listed by names and a Roman numeral. So if I say cranial nerve 1, it could also be listed as CN1, and the 1 is usually in a Roman numeral. That's basically talking about the olfactory nerve. So what does it do? Now you got to keep in mind one other next thing. There's more, of course. Some of these cranial nerves only take up sensory information, bring it up into the brain. Some of them only send out motor information, in other words, motor instructions. They might cause certain muscles in the face or shoulder, whatever, to move. Some of them are what is referred to as mixed, meaning that they take care of some sensory actions, taking up sensory input, but they also then send out some motor activity, moving muscles, etc. So it is important that you go over the olfactory, which is all sensory, optic, all sensory, and now we start getting into ocular motor. The ocular motor, the trochlear, okay, and the abducens are all found helping to control the eye muscles. The trigeminal nerve, tri meaning 3, you'll see in a minute. This is cranial nerve 5 and plays an important role in some of the facial movements and muscle movements and other things. The facial nerve does the same thing. The vestibular cochlear nerve is mostly sensory because it takes up the vestibular information, which is the sense of balance, etc., from the inner ear, as well as the cochlear part, which is the processing of sound. Okay. The glossopharyngeal, well, glossio involves speech. Pharyngeal, these are all involved with functions around tongue and the pharynx. Now, the vagus nerve is very special. This is referred to as cranial nerve 10, but the vagus nerve plays a key role in passing parasympathetic innervation to a variety of organs in both head, neck, thorax, and upper part of the abdomen. Okay? When we get into the autonomic nervous system, you're going to be reintroduced to the vagus nerve and how absolutely essential it is for the proper homeostasis for the person. The accessory nerve is mostly motor, and the hypoglossal, I believe, also is mostly motor. But we'll go through this a little bit more in depth. Olfactory nerve originates in the nasal cavity. The optic nerves originate from the retina of the eyes. Now, originating from the midbrain, and this is part of the points also, a lot of these nerves do not attach to, let's say, the frontal lobe of the brain. This is all stuff that's going into a variety of the midbrain, pons, and medulla oblongata. All right. So originating from the midbrain, you have the ocular motor nerve, which is cranial nerve 3, trochlear nerve, cranial nerve 4. Originating from the pons, that's the trigeminal nerve, cranial nerve 5, abducens, cranial nerve 6, and the facial nerve, cranial nerve 7. Originating on the pons medulla border, vestibular cochlear nerve, which is cranial nerve 8. Originating in the medulla oblongata, the glossopharyngeal nerve, cranial nerve 9, vagus nerve, cranial nerve 10, accessory nerve, cranial nerve 11, and hypoglossal nerve, cranial nerve 12. And you'll see all of these. So if you notice underneath, and again, I don't want you to worry that much about right here. Having this as the optic nerve, understanding this is the olfactory nerve, that's a beginning. But you should know what these individual ones do, okay? A lot of these are involved with monitoring stuff. Some of it you're conscious about. You know, it's like when I make a smile or something on my face. But others, you don't have to think about it. You know, eyes turn to the left, eyes turn to the right. Okay. These are things that we do unconsciously, otherwise known as involuntary. Now, there are a variety of cranial nerve tests, and they are mentioned, and these are tests for olfactory, oculomotor, trochlear, abducens, etc. These I've asked to skip because we don't have always the chemical tests available or the tests for the glossopharyngeal, vagus accessory, and the hypoglossal nerve. It's interesting to read over them. You may see where in some cases, a basic clinical review gives you a lot of information. I believe I included in one of the videos, and I don't know if it was for the lecture or the lab, but there is a doctor that says, you know, he teaches what is kind of called disaster medicine, or, you know, when there's adverse situations. You have a patient come in and he basically asks a bunch of different questions. And by asking the questions, he can actually do a test for pretty much most of the cranial nerves, some of the spinal nerves, and I encourage you to look it over. Okay. You know, even certain things where he's asking the person just to say their name. Well, they can hear it and then they're going to respond to it. It gives him an idea about what nerves are functioning, etc. Now, I do want you to review skull openings for the cranial nerves and recall the terms foramen, fissure, and canal in the skull. You have to go back to the earlier part and review that. Using an open skull, the cranial cavity, the neural cranium, locate the following: the cribriform plate, which is part of the opening for the olfactory nerves, the foramen rotundum, which is partially cranial nerve 5, the foramen ovule, which is partially also cranial nerve 5, the jugular foramen, which is cranial nerves 9, 10, and 11, the optic canal, which is the optic nerve, cranial nerve 2. The superior orbital fissure, which takes care of cranial nerves 3, 4, and 6. The internal acoustic nidus, which covers cranial nerve 8. The hypoglossal canal, which covers cranial nerve number 12, and the styloid mastoid foramen, which is partly cranial nerve number 7. And you can see how these are all arranged here. That's what I want you to take, review this image, and review this image to see clearly how these are. Now, you might sit there and say, this is a lot of minutia. No, it's not. Patient comes into you, had a very severe accident, and there was damage to one side of the face. Let's say the damage included compression crush injuries right around here. They might say, well, how come I'm somewhat awake, but I don't feel certain things on this side of the face, etc. I don't feel certain, because some of that compression, crushing could have actually cut these nerves, severed them, or damaged them. or they're reporting tremendous pain, and it's basically because of the crushing injury has now put a lot of pressure on those nerves. Case in point. Well, it seems like we've gone through a lot here. Okay, here's what we're going to do. Review the exercises 14.1 to 14.3. Omit 8b, which is 15.8b, 15.9, and 15.6. That's the one with the biopack we don't have. Do the rest of 15. As a matter of fact, to help you fill out on those pages any particular information you may have. Then when you get to your review sheet, you can put the information in. It'll help you. As always, keep the originals of your review sheets. Put the ones you want to submit to me in PDF format. Also complete whatever images of microscopic slides that you're going to see, and any models, etc. Also any dissection information that include in the PowerPoint and in the videos. Those will still be held to you for the subsequent lab practical exam number 2. Okay? Remember that you need to submit your worksheet by Friday, 5 p.m. Also review your lab videos and the lab manual notes for the final lab, which will be on the special senses, because we want to get you prepped up to do well on that lab. And after special senses will come lab practical exam number 2. I hope you have a great day.